A very good day to Dr. Suraj and Dr. Alicia. So today we are going to present our bio, our first bioinformatic assignment, which is called BLAST, known as Basic Local Alignment Search Tool. So here is our member and what are they in charge of which part. So here's our topic outline. We will start with the introduction, different version of BLAST, uses of BLAST, method used in BLAST, explain about BLAST, and finally conclusion. So I will start with the introduction. So what is sequence alignment? So sequence alignment is a process of comparing and detecting similarities between biological sequences. It can be used to capture various facts such as common structural function or evolutionary descendant of aligned sequences. The sequences can be in the form of protein, which is monomer amino acid, and DNA and RNA sequences, which is in the form of the nucleotide bases A, T, G, and C. So the example of tools that are commonly used for sequence alignment is EMBL, GenBank, UniProt, KB, etc. So here's a very brief historical trend about sequence alignment. So in 1970, the presentation of dynamic programming algorithm by Tiedemann and Wunsch, and in 1981, the Smith-Waterman local pairwise alignment is discovered. BLAST was introduced in 1994 by Ground and his colleagues. So there are two types of alignment. Sequence alignment refers to be the local alignment. It uses the sequence with more similarity, and it is commonly used for dissimilar sequence with suspected similarities. And next we have global alignment. It is the sequence alignment that involves the entire length of sequence, and it is commonly used for matches identical sequences. So this is our topic for the presentation. So BLAST is known as Basic Local Alignment Searching Tool. It's one of the widely used bioinformatic programs in sequence searching. So it is developed by NCBI, and it is the algorithm or program that performs sequence comparison and identification. And the sequences are in the form of amino acid, which is the monomer of protein, and or nucleic acid, which is the RNA or DNA. Yeah, I will pass it to my friend for the next part.
Hello everyone, I am Muaz and I will present about the different version of BLAST program. There are a few BLAST programs which are different in database and query. What does it mean with query? A group of applications used to create alignments between nucleotide or protein sequence. The BLAST program used in certain and specific findings regarding protein and nucleotide sequence. Next, I will show the five types version of BLAST program. As you can see here, there are 5 types of version of BLAST program, which are BLAST N, BLAST P, BLAST S, T BLAST N, and T BLAST X. The first one is BLAST N. The database is nucleotide and the query is nucleotide. The use of BLAST N is cross-species sequence exploration, and the second one is annotating genomic DNA. The second program is BLAST P. The database is protein and the query also protein. And the use of BLAST-B is identifying common regions between proteins. The third one is BLAST-X. The database is protein, the query is nucleotide translated into protein, and the use of BLAST-X is finding protein coding genes in genomic DNA. The next one is T-BLAST-N. The database is nucleotide translated into protein, and the query is protein. The use of T-BLAST-N is mapping a protein to a genomic DNA, and the last one is T-BLAST-X, which the database is nucleotide translated into protein, and the query also same, nucleotide translated into protein. And the use of T-BLAST-X is cross-species gene prediction at the genome or transcript level. There are a few and lots of more of the use of the BLAST program. I will pass to the next presenter. That's all from me. Thank you.
Hello, my name is Mama Aziz. So now I will explain on the uses of the BLAST. So the first one is BLAST can be used for DNA mapping to a non-chromosome and also for comparison. By using BLAST, we can see the position and location of the query sequence in relation to the hit sequences. And from this, we can make comparisons between nucleotide and protein sequence and also estimate the statistical significance of matches. We can also identify the functional and evolutionary links between the sequences, as well as assist in locating the members of gene families. The second use of BLAST is it can be used for homology searching. So homology means a common ancestry and similar structure to predict a gene using alignment of protein. BLAST can be used to characterize new determined sequences, and it can also detect homologous proteins or genes by looking for statistically significant similarities that imply the common ancestry. Therefore, BLAST can present an overview of homology inference from significant similarity and also introduces other units that can provide further details on the effective ways of discovering homologs. The third one is species identification. It will enable for the simple uploading of DNA sequences by intuitive user interface to identify the species. From this, researchers and also students can upload or paste the DNA sequences and then query them. So from all of these sequences, we can compare them with a known database to identify the species. The last one is BLAST can be used to generate phylogenetic tree. Phylogenetic tree is a diagram that represents the line of evolutionary descent of different species, organisms, or genes from a common ancestor. So by using BLAST, we can find and identify similarity with other sequences. Sequences that are matched with each other can develop phylogenetic relationship between the sequences.
Hi and a very good morning. So I am Tanim. So I will present about the methods used in BLAST. So basically there are 6 methods used in BLAST. So for the first one is statistical significance. So to make sure that extracting sequence from a purely numerical measure of similarity is very difficult. So these values are calculated and quoted in the results generated by database searching algorithm. So for the next method is e-value. So for the e-value, statistical measure of the significance of sequence. It is the statistical measure of the significance of a sequence similarity match. So to put it simply, a lower e-value indicates more significant match. Next is algorithm. So algorithm will break the query sequence into smaller pieces and searches for local alignment with the database sequences. So for the next method is substitution matrix. So substitution matrix is used to score aligned positions in a sequence alignment. So basically, there are 2 commonly used matrices, which is PAM, which is percent accepted mutation, and BLOSSOM, which is block substitution matrix. So basically, PAM and BLOSSOM has their own function and differences. for the PAM is based on explicit as a evolutionary model, and for the blossom is based on empirical frequencies. So higher PAM numbers to detect more remote sequence similarities, while lower blossom numbers to detect more remote sequence similarities. And for the next one is gap penalties. So gap penalties is to penalize the introduction of gaps in an alignment. They are using two separate negative scores, which is gap opening penalties and gap extension penalties. So gap opening penalties is a large penalty for introducing a gap, while gap extension penalties is a much smaller one, which is used to extend an existing one of the sequence. And for the last method is scoring matrices for DNA and protein comparison. For DNA comparison, a height score is given for a mesh between bases and 0, while the negative score, by meaning to say, it is defined by mismatch. So a mesh between A and not A is scored as a mismatch under all circumstances. And for DNA, for protein comparison, it will determine the significance of a match between the query sequence and a database sequence. And it also uses PAM and blossom matrices. So that's only for my part. I will pass to the next presenter.
Okay, Assalamualaikum. My name is Cik Muhammad Farhan bin Cik Mat. So now I will explain about the BLAST-P. So BLAST means the basic local alignment search tool, and the P here means protein. So basically it is a tool which is used to compare between the protein sequence against the protein database. So it uses the algorithm of the BLAST, which is to identify the similarities and differences between two or more protein sequence, and it allows researchers to compare a protein sequence of their interest with a vast collection of proteins. So BLAST-P can be used to identify conserved domains, catalytic residue, and other functional motif in a protein sequence. Okay, so now we move to the pros and cons for the BLAST-P. So firstly, for the pros for the BLAST-P is that it is a very powerful and well-known tool to identify domains and homologs of the protein sequence, and it is a very useful tool when we have to analyze a large amount of the data set. And as the BLAST-P can accurately produce result with a high sensitivity and specificity, means that it can effectively produce only minimal false positive. And lastly, it can reduce time and researches by providing evolutionary relationship and similarity amongst the proteins. So next we move to the cons for the BLAST-P. So here we have sometimes it may not be accurately predict the protein function or evolutionary relationship because it relies on the sequence similarity based approach. So next, if the input query sequence is not filtered properly, or or the protein database is poorly curated, it may prone to produce false positive result. And next, cons is the not well in identifying divergent or newly evolved protein. And lastly, the output result may be difficult to interpret. For example, like when dealing with the large dataset or closely related homologs. So that's all from me. Thank you so much.
BLAST, which is known as Basic Local Alignment Search Tool, is a widely used bioinformatics tool for comparing nucleotide or protein sequences against a database of known sequences. BLAST has revolutionized the field of bioinformatics by providing an efficient and accurate method for identifying similarities between sequences. In conclusion, BLAST is an essential tool for many bioinformatics applications, including sequence annotation, phylogenetic analysis, and functional prediction. Its ability to rapidly identify similarities between sequences has helped researchers to understand the evolution and function of genes and proteins across a wide range of organisms. However, it is important to keep in mind that BLAST results should always be interpreted with caution and verified through additional experimental and computational analysis. And for the last slide is my team references throughout making this video assignment. That's all from us. Thank you.